Hello everyone, I'm Priyanka and today we will discuss the distillation process. Distillation is the process of converting liquid into its vapor by heating and reconverting it again into liquid by condensing the vapor. It is the method of separating liquid which differ in their vapor pressure. The distillation process is carried out in an apparatus which consists of still in which volatile material is boiled, a condenser in which vapor are condensed, a receiver in which distillate is collected. Further, the distillation types are, are as follows. First, simple distillation, distillation under reduced pressure, fractional distillation, steam distillation, destructive distillation. Firstly, let us talk about simple distillation. It is the process of converting a liquid into its vapor in a distillation still, transferring the vapor to another place and condense it again into liquid. As the given as the figure given below shows a simple distillation process on laboratory scale. Apparatus used for the laboratory scale. It consists of a distillation flask with a side arm sloping downward which is connected to the condenser. The condensed vapor are collected in a flask called receiver. The whole apparatus is made up of glass. The distillation flask should be of such a size that it can contain half to two-thirds of the liquid to be distilled. The thermometer is fitted in the distillation flask to note down the temperature at which the vapors are distilled. Bumping is avoided by adding small piece of porous pod before distillation. Talking about the application of simple distillation, it is used for preparation of distilled water and water for injections. Many volatile oils and aromatic water are prepared by simple distillation. Organic solvents are purified by distillation. Many many official compounds are prepared by distillation, for example, spirits. Next, we have distillation under reduced pressure. The liquid boils when its vapor pressure is equal to the atmospheric pressure. The boiling point of the liquid may be lowered to the desired temperature by reducing the pressure on its surface. The figure given below shows the distillation under reduced pressure on laboratory scale. Talking about the apparatus used for the laboratory scale, it consists of a double neck distillation flask known as Gleason flask. In one of its neck, a thermometer is fitted, and in the second neck, a capillary tube is fitted, which prevent bumping of the heated liquid. The capillary tube should be so lined as to permit only a slow stream of bubble which can be controlled with a pinch cork. Thick wall glass apparatus with interchangeable standard glass joints are used for vacuum distillation. The Gleason flask is connected to a receiver through condenser. Vacuum pump is attached to the receiver to attain desired degree of vacuum. Heating in Gleason flask is not started until the desired vacuum have been attained. Talking about the application, it is used for the concentration of extract containing thermolabile material in order to prevent their destruction. It is used for separating substances which undergo decomposition when heated under normal atmospheric pressure. Next, we have vacuum still. The vacuum stills are employed for distilling substances under reduced pressure on a large scale. A vacuum still is generally made up of stainless steel or any other metal which can withstand high vacuum. The still is connected to condenser. The vacuum is created by means of vacuum pump. The given figure shows the vacuum still. Vacuum still is fitted by attaching a pipe to a tape in the lower part of the hood and the pump is started. The other end of the pipe dip into the liquid to be distilled so that it can be drawn into the still. The observation window is the hood in the hood is very helpful to the operator to see the progress of distillation and also the level of the content of the liquid to be distilled. Two receivers are generally attached to the condenser in order to collect the distillate without stopping distillation. However, they may be used alternatively by a suitable arrangement of corks. Talking about the application, distillation of substances that have a high boiling point at atmospheric pressure. Distillation of thermolabile substances that get damaged by a high temperature. Removal of last traces of volatile solvent. Next type of distillation we have is fractional distillation. Distillation of miscible liquids. When a substance is dissolved in a liquid, the vapor pressure of the liquid is lowered. When two miscible liquids are mixed together, each will act as solute or solvent for the other. So, when a mixture of two such liquid is heated, the vapor pressure of each is lowered. The pressure exerted by each liquid in the mixture is known as partial pressure. The liquid boils when the sum of the partial pressure is equal to the atmospheric pressure. The vapor arise from two miscible liquid at boiling point is richer in the component exerting the greater partial pressure. Talking about the apparatus for the laboratory scale, the apparatus used for the laboratory scale is shown in this figure. Fractionating column is fitted between the distillation flask and the condenser. Fractionating column is used for continuous separation of two miscible liquids. Long fractionating column is used in the mixture where the boiling point is quite close to each other, and short fractionating column is used in those cases where there is a considerable difference in the boiling point of the mixture of miscible liquid. The figure given here shows the fractional distillation on the laboratory scale. In the fractional distillation, The mixture of miscible liquid is heated in the still. The vapor formed are allowed to pass through the fractionating column where a part of vapor is condensed and while returning to the still come into a intimate contact with the rising vapors resulting in further fractionation of the liquid being distilled. The liquid with high boiling point is condensed first and vapor become richer, with the liquid having the lower boiling point which get condensed in the condenser. Talking about the application, alcohol is purified from the mixture of alcohol and water obtained from the fermentation tank. It is used for the separation of miscible liquid such as alcohol and water, acetone and water, chloroform and benzene. That's all for today. Thank you and have a nice day.